Bismillahirrahmanirrahim. We start another paper. This is the second video on the paper 4-2, November 2021. And the next three questions, I've done the first three questions in the video 1, so this is the second video. We start with question number 4. A. Genome-wide association studies find links between single nucleotide polymorphism SNPs and phenotypic features such as human diseases. SNPs or SNPs are points on the DNA that vary in the population because of DNA base substitutions. Please understand that. DNA base substitution. So in certain people, in a majority of people, there's a certain base sequence, and in a few people, there's this base substitution takes place. A genome-wide association study investigates the effect of genetic variation on a disease. A large number of people with the disease and a large number of healthy control individuals provide DNA. Microarray chips are used to identify the genotype of each individual at many SNPs, SNPs. The Wellcome Trust Case Control Consortium (WTCCC) study was an important genome-wide association study. The study used a microarray chip that identified each person's genotype at 500,000 different SNPs, SNPs. The study looked for links between SNPs and 7 different diseases. For each disease, 2,000 people with the disease were tested. 2,000 people had a certain disease, they were tested. Their results were compared with the result of 3,000 healthy control individuals. So 2,000 people were diseased and 3,000 were the healthy control individuals. Question part 1. Outline how microarrays are used in the analysis of genome. Now that's a very direct question. Either you know it or you don't know it. You can't be guessing this. Then explain why bioinformatics was important to the WTCC study. Now you can see what is a single nucleotide polymorphism SNP. There are positions in a genome where some individuals have one nucleotide, example a G, and others have a different nucleotide, example a C. So you can see this person A has an A here, this person B has G here, and this person C has T here. So three different people just have a different base in a certain place in the position. So normal carrier disease, sometimes it doesn't have any effect, sometimes it results in the person becoming a carrier, sometimes. Now you can see another of these examples. This is another example, common genotype B variant genotype. This is the SNP site, and this results in a protein, and this results in an altered protein. Then another example is here, individual 1. Now just pause the video here, have a look at it, and see what is the difference that they are showing you in this situation. So there is only one base which is different. So individual 1, you can see instead of TA, there's CG. In individual 2, instead of CG or CG, it's again a CG. Then individual 3, TA, then individual 4, TA, CG, then individual 5, CG, TA. So there are these different 6 individuals in which they've shown you their genotypes. Not the genotypes, but the base sequences in the DNA. As you can see here in the microarray question, what is a microarray? It's a very direct question. Either you know it or you don't know it, as I said earlier. You obtain single-stranded DNA. So you take the DNA of a person and you ensure that it is not no longer double. You break the hydrogen bonds between the two DNA strands. So you obtain single-stranded DNA, which is also called ssDNA. So you obtain single-stranded DNA. Then you label the DNA with fluorescent dye or a tag or a label. Then you probes on the chips. On the chips, you have these little circles and you have these probes on it. Probes means you have these different DNA. So you could have A, T, 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 or something like this, or A, C, C, C. So you have these probes on it, which is the microarray, and each probe is unique to a particular gene or an allele or a SNP. Then the DNA is added, which binds to these probes. So you know, I mean, if it is there is a T, 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 A, only that will able to bind to this. If there's not this sequence, then of course it won't bind to it. So DNA binds or hybridizes to the probes. Then wash off the excess DNA, the one which doesn't hybridize, you just wash that off. Then you view under the UV light or a laser scanner or a high resolution digital camera. and the fluorescence indicates the presence of the gene, or if it's present or not. Please don't say gene expressed, because if the gene is expressed, then you would see proteins. Here, no proteins are seen here. Gene is not expressed, so reject gene expressed. That would be a reject, means a large number of students would have written that. Then bioinformatics. What was bioinformatics? There's a large database, the use of the database to find which probes have hybridized, which ones have hybridized with the probes. Then there's a fast and accurate and efficient method. Then you need software and algorithms for it. You need statistical analysis of it. And you have so many data points, something like huge sums of data points that you have. Now, coming to the B part of the question, figure 4.1 summarizes the results for 3 diseases in the WTCCC study. The 22 human chromosomes and the X chromosome, chromosome 23, are shown. Chromosome location with SNPs that are associated with a disease at a statistically significant level greater than 5 arbitrary units. So you can see here, this is 5, this is 5, this is 5 arbitrary units. So are associated with disease at a statistically significant level greater than 5 are shown in black. Now you can see all these. This is for rheumatoid arthritis, this is for type 1 diabetes, and this is for type 2 diabetes. And chromosome numbers are given, they're all the same in the 3 diagrams. Then it says identify the chromosome that contain SNPs that have a high level of association with both rheumatoid and type 1 diabetes. So first you circle this, that you're going to look at these. Then you look at the arbitrary units. How many of there are here more than 5? So 5, you can see here, this one is more than 5 here, and this one is more than 5 here. Here also this one is more than 5, and this one is more than 5. And we have to see these two. So the answer to this was 1 and 6. So 1 and 6, you can see here, this is the 6 here, this is the 1 here, this is the 1 here. So 1 and 6 have a high level of association with both rheumatoid and type 1 diabetes. So as we go ahead, then it says, with reference to figure 4.1, compare the genetic basis of the 3 diseases. And that is for 2 marks. So the genetic basis of the 3 diseases. Now let's look at this figure here again, because it says reference to figure 4.1. So let's go through this again, and then we will discuss this. First you need to do is, you've read from the question that it is anything is more than 5 arbitrary units. So just look at the ones which have these, which are more than 5 arbitrary units. So use chromosome 1, chromosome 6 here, which I have just underlined. Then here chromosome 1 here, then 6 here is more, then 12 here is more, then 16 here is more, then here we have again 16, and here we have 10 here. So now when they asked you, in the question they asked you, compare the genetic basis of the 3 diseases. Now the first thing which you come to your mind is none of them are associated with the sex chromosome. So the sex chromosome is the same in all 3 of them, and that doesn't have anything more than 5 arbitrary units. So the 5 one here, 5 is here, and 5 is here. So anything more than 5 was the one which was bad. So all diseases are autosomal and not associated with the X chromosome, which means they are not sex linked. Then all diseases are associated with SNP on chromosome number 6. So you can see nearly all of them are associated with the on chromosome number 6. Here also we have 6 is a little higher here as well. So most of all the diseases are associated with SNP on chromosome 6. So that was one common thing. Then type 1 and type 2 are associated with 12 and 16. So type 1 and type 2 are associated with 12 and 16 here. And here we also have 12 a little higher, and 12 and 16 here as well. So type 1 and type 2 with 12 and 16, but not rheumatoid arthritis. So rheumatoid arthritis is left out here. Then type 2 diabetes and is associated with chromosome 10. So type 2 diabetes is chromosome with more with chromosome number 10. So here is the one which is a little higher. So type 2 diabetes was associated with that. Then arthritis and type 1 have a higher level of association with each other than with type 2. So these and they both have a slight association with each other rather than with type 2 diabetes. Then rheumatoid arthritis has the fewest number of SNPs. So if you see at this one, rheumatoid arthritis only has 1 and 2, while here we have 1, 2, 3, and 4, while here again we have 1, 2, 3, and 4. So you can see this. So now look at the Marx scheme. The Marx scheme is very clear. All you had to do, this is nothing which you had studied before either in the school or your teacher had taught you this, but it is how you understand a question and how you then comprehend it and write down the answer. You must take time in reading the question and you must take time in answering it in a very, very befitting manner. Now, just going through the Mark scheme, all diseases are autosomal, not associated with X chromosome, or you could have said not sex linked. All diseases are associated with SNP on chromosome 6. Type 1 and 2 diabetes SNPs are associated with chromosome 12 and 16, but not rheumatoid arthritis. Then type 2 diabetes SNP associated with chromosome 10, but not type 1 and the rheumatoid arthritis. Then arthritis and type 1 diabetes have a higher level of association with each other than with type 2 diabetes. Rheumatoid arthritis has the fewest number of SNPs. Type 1 and type 2 have more SNPs than rheumatoid arthritis. So basically, it's nothing something which you had studied. You had to just read the question and you had to have your biological mindset in order, and you would have been able to figure this out if you can understand the question and if you can comprehend the question. Then coming to the C part of the question, individuals can choose to have their DNA analyzed on a microarray chip to predict their risk of developing different diseases. Outline the social and ethical consideration of this type of DNA analysis. Well, this is the biggest question which they always have given you in the syllabus. DNA analysis is not available for everyone. Then lifestyle changes, example, lose weight, stop smoking, take exercise. Early treatment can be done if, of course, people have done this. So the good is, of course, then you can take changes. You can take early treatments. You can allow people to plan, organize, or uh, take out medical insurance, or you can make certain decisions, gift property to children, retire early. Then you can decide whether to have children or not if you feel you have some genetic disorder. Now, of course, the bad side is that the results that may cause anxiety, stress, panic, depression, or if you're clear of it, then it reduces the worry, because if you don't have any issues, then of course you are lucky. The results may affect the ability to get insurance or credit or jobs, and predictions might not be so accurate. You see, later on they might decide, oh my God, we used to do this DNA sampling and this was not very correct, and these were some faults in it, or there were some mistakes in it. So social and ethical considerations, you've got to come with the goods and the bads. So DNA analysis is not available to everyone. Just going through the points, DNA analysis not available to everyone. Lifestyle change. I mean, why do you say lifestyle changes? Because if you get to know that you have a heart disease or you're prone to heart disease, then you can make lifestyle changes, lose weight, stop smoking, take exercise. And then if you know there's something which you can be treated, so you can get early treatment for that, then allows people to organize, plan, organize care, appoint power of attorney, take out medical insurance, make a will, gift property to children, retire early. All these are different lifestyle decisions that you could make. Then decide whether or not to have children, because or you can do a therapeutic termination if you know the child is going to have a certain illness. Results may cause anxiety or stress, or you can say the other way around, will reduce the worry if you know you have nothing to worry about. Results may affect ability to get insurance or credit or jobs. An idea of predictions may not be accurate. Yes, it could be this is not some sort of a very foolproof test, and maybe in the years to come they realized they were making some wrong predictions. Now, coming to question number 5, biodiversity can be considered at 3 different levels. Figure 5.1, 5.2, and 5.3 are images that show these 3 different levels of biodiversity. So figure 5.1, you can see all these grass, and then you can see all these goats in 5.2, and then you can see all the water and the different grasslands, so that's 5.3. So 5.1, 5.2, and 5.3. Then let's look at the question. Describe the level of biodiversity most clearly visible in each image. So figure 5.1, what do you see? You see species. And figure 5.2, you see genetic. And then figure 5.3, you see habitat or ecosystem. You see water and you see all that. So levels of biodiversity most clearly visible in each image. That was a little difficult, I feel, because you could see if you look at the diagram, why is it? This is species, grass and flowers. Then 5.2 is genetics. You can see the same species, but their genetics are different. Their colors are different. And this, of course, you can see the sky and the water and the land and the pond. So that would be habitat or ecosystem. Then it says prairie strips are restored habitat areas planted with native grasses and wildflowers. Prairie strips are usually located between or around edges of fields of maize or soybean. Students in North America wanted to investigate biodiversity along a prairie strip. Outline how the students could measure biodiversity of plants and insects along a prairie strip. So basically, how would you investigate plants? And that is, of course, you use the quadrats and you use transects, and you can use the Brown blanket scale. Insects, of course, you would use pooters, or you could use even the mark release recapture, and then you would use the identification keys to decide which one are which this thing. Then mathematical methods and statistical tests help to summarize and compare biodiversity. Name the mathematical method or statistical test that the students can use to estimate the total biodiversity of a prairie strip. Now you know that is Simpson's index of biodiversity. So that is Simpson's index of biodiversity. Simpson's index of biodiversity. Then to test whether plant biodiversity is associated with insect biodiversity. Now which one is that? That is the Pearson's linear correlation. Pearson's linear correlation. Pearson's linear correlation, or you could also say the Spearman's rank correlation. So the Spearman's rank correlation you could also have said, or the Pearson's linear correlation you could have also said. Then read the question very carefully. See, prairie strips have environmental and economic effects. Scientists have made long-term measurements of the environmental effects of prairie strips. The scientists found that when prairie strips formed 10% of a crop field, soil erosion decreased by 95%. Mineral loss from the fields decreased by 90% for phosphorus compounds, and mineral ions from the fields decreased by 85% for nitrogen compounds. Crop plants need phosphorus and nitrogen to grow. Evaluate the economic effects of prairie strips on farming. So we look at the negative impacts would be, of course, if the prairie strips occupied areas, so then there would be less area available, so smaller crop area, so lower yield. And then we had the negative point was prairie stripped insects may feed on these crops. And of course, this would result in less profit or less income. Say, you have to read the question very carefully. Mineral loss from the field decreased by 90%. Mineral loss from the field decreased by 85% for nitrogen compounds and phosphorus compounds. So the positive factors are the soil will be more fertile, so higher yield. Less fertilizer needs to be used. We need less phosphorus and less nitrogen compounds. So if we don't have to spend money on fertilizer, this is going to reduce our expenses. So the art of paper 4 is to read the question many times to understand the question, and then of course use your biological mindset and your biological knowledge to understand. If we don't have need fertilizer, of course we'll have less money will be used on fertilizer. So the expenses will be reduced. So less money spent and more crop yield and more crops, so more money. And coming to question number 6, ATP is needed for many metabolic processes in living organisms. Describe the properties of ATP that make it suitable for its role as a universal energy currency. Very easy, it's come in many papers. Is water soluble, easily transported around the cell. ATP loses the phosphate group. It is hydrolyzed by ATPase. So ATP is a stable molecule, releases energy immediately in small packets, and can be recycled or regenerated. So very easy points, and I'll just go through it once again with you. Suggest why ATP is needed for protein synthesis. Why is ATP needed for protein synthesis? Unwinding the DNA, activating the RNA nucleotides. We need it for mRNA synthesis, the peptide bond formation between amino acids. And then we need energy for post-translational modification, or moving the ribosomes along the mRNA, or the movement of the mRNA from the nucleus. So all these were acceptable points for part 1 and part 2. Just so that you learn the wordings again. So ATP is water soluble, easily transported around the cell. ATP loses the phosphate group, hydrolyzed by ATPase. ATP is a stable molecule, release energy immediately in small packets, and can be recycled or regenerated. Then why did we need it for protein synthesis? You remember the DNA has to unwind, and then the mRNA has to copy it. So the mRNA copies this template strand. So unwinding the DNA, activating the RNA nucleotides, then mRNA synthesis, then peptide bond formation between amino acids, and then post-translational modification. Then figure 6.1 is a diagram of the mitochondrion, diameter, length, A, B, C, D. Then what does it say? Complete table 6.1 using letters A to E from figure 6.1. Each letter may be used once, more than once, or not at all. So then we come on to the site of the Krebs cycle. Where is the Krebs cycle going to take place? That's going to be at A. And then phospholipid bilayer impermeable to hydrogen ions can be E, or it can be D. That was also acceptable. And the site of translation would be C. So let's go back and look at it. The site of the Krebs cycle, you know, where is that going to take place? That had to be A, because that's the matrix of the mitochondrion. Then a phospholipid membrane impermeable to hydrogen ions was E or D. E is the inner membrane, or D, which is the outer membrane. And the site of translation again was C, because that's the 70S ribosome which they have shown you, and that is C. So the site of translation, and because they haven't shown you the loop of DNA, but they've shown you something like that, looks something weird. But these little dots are the site of the ribosome, the 70S ribosomes, where the site of translation is going to take place. Coming to part 2, assume that the mitochondria is a cylinder. Calculate the surface area of this mitochondrion. Use the formula surface area of cylinder is 2 pi r square plus 2 pi r h. Show your working. So this is the working that was expected of you, and you were supposed to give me the answer. Either you gave me 12.7, or you gave me 12.68, or 12.69 was also acceptable. Then coming to the next part of the question, the inner membrane of the mitochondria has a much larger surface area than the outer membrane because of the presence of cristae. Different cell types vary in the number of cristae present per mitochondrion. Cardiac muscles have mitochondria with a very large number of cristae. Suggest and explain why cardiac muscles have mitochondria with a very large number of cristae. Now, you know the outer membrane is like this, the inner membrane is folded. So this increases the surface area, because what do you have to have here? You have to have the enzymes here, the multi-enzyme complex. And then you have to have the ATP synthase. So you have to more of these. So you have this here, then you have this here again, then you have this here again, and then you have the ATP synthase. So more the foldings, more of these sets of these proteins can fit in there. So basically you have to understand that more crystal results in more more ATP synthase. Cardiac muscle must undergo continuous contractions, so it needs to generate more ATP. It needs to release more energy. I said generate more. You see, generate more ATP. You can say produce ATP, but you can't say produce energy. So energy is released, but ATP is produced. You've got to understand that. So more cristae, more ETC, and more oxidative phosphorylation, more ATP synthesis. And cardiac muscle has to work all the time. From the day you're born till the day you die, the cardiac muscle, poor thing, cannot even rest. So you have to give me this. Why would there be more cristae? So more ATP synthesis, more ATP synthase molecules can then fit in there. Thank you for watching. We finished question 4, question 5, and question 6. In the next video, we will do the last two questions, which is question 7 and 8. Thank you very much.